Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. You can find Let's Talk Micro on Apple Podcasts, Google Podcasts, Spotify, Amazon Music, TuneIn Radio, Stitcher, Overcast, Good Pods. Wherever you find your podcast, you can find Let's Talk Micro. As far as social media, I am on Instagram as Let's Talk Micro, no apostrophe, and on Twitter as Let's Talk Micro 1. I am also on LinkedIn as Luis Plaza. So go ahead and subscribe to the podcast. Go ahead and leave some feedback, any topic suggestions, any feedback and suggestions. They're always welcome and appreciated. And if you haven't listened to the previous episode of Let's Talk Micro, go ahead and do so. This was another educational piece. I mentioned that as the pandemic hit, educators had to adapt on how to teach their students remotely. How can we teach microbiology when the students are not present doing the hands-on work, acquiring those hands-on skills? So I had one episode where an instructor, a microbiology professor, he managed to teach his students streaking remotely. They were inoculating plates, they were streaking them. And in this last episode, I had Heather Townsend and Catherine Leonetti, and they are two college professors, and they implemented two ways of teaching microscopy remotely. So it's a great episode. They talked about what worked, what didn't, how they have incorporated this style of teaching into their current, methods. So they learn a lot, and they are currently implementing this method in their class. So it's a great episode. Go ahead and check it out. So, on today's episode, Dr. Paul Hergenrother from the University of Illinois, he joins the podcast to talk about Fabimycin, which seems to be a promising candidate for, an antibiotic, especially for gram-negative bacteria. He talks in the podcast about how Fabimycin works, which it just, inhibits Fabi, which is an enzyme, a key enzyme in fatty acid biosynthesis. He talks about how Fabimycin did against known pathogenic gram-negative rods. So he talks about that. He mentions that Fabimycin, one of the things that makes it very interesting and promising is that it only kills, the pathogenic bacteria, and it doesn't kill the commensal bacteria, that normal flora that we have, which those of you that are microbiologists, you know how important it is to have that normal flora. So many things, he mentions that there hasn't been a drug that targets gram-negative bacteria, that there hasn't been a new one in the market in many years. So he discusses why this is so. So in summary, we talk about this promising drug, which is Fabimycin, how it works, its target bacteria, and what are the next steps. Really good information. So let's go ahead and check it out. So on today's episode, I have an interview and I have a guest with me to discuss an article titled An Iterative Approach, Guide Discovery of Fabi Inhibitor Fabimycin, a late-stage antibiotic candidate with in vivo efficacy against drug-resistant gram-negative infections. This was published in the Journal of the American Chemical Society Central Science on August 10 of 2022. So with me, I have Dr. Paul Hergenrother. Doctor, welcome to Let's Talk Micro.
Great to be here. Thanks so much for the invitation.
Definitely my pleasure. So let's go ahead and start with an introduction.
My name's Paul Hergenrother. I'm a professor of chemistry here at the University of Illinois at Urbana-Champaign, and I've been here for about 21 years, leading a team of graduate students and postdocs and undergrads, and looking at these important problems in modern medicine.
Well, thank you for your work. So I'll be asking questions as we go along, but let's go ahead and can you do an overview of this article and what's it about?
Sure. Basically, we've been studying a certain type of bacterial pathogens for a while. These are called gram-negative bacteria, which tend to be more challenging to kill with antibiotics, and that's something I'm sure we'll get into in this podcast. And we've been looking for new approaches to develop antibiotics for these pathogens. And in this particular article, we find a novel antibiotic that we call Fabimycin that is really interesting in its ability to not only kill these gram-negative bacteria, but to specifically kill pathogenic gram-negative bacteria, and while sparing bacteria that are called commensal or so-called good bacteria in the gut. And so that is, I think that's something that's gotten a lot more recognition lately, is the deleterious effects of antibiotic therapy. And so with this compound and other analogous approaches, the idea is really to hit the bacteria that are causing the infections and not other bacteria.
Okay, yes, definitely. I mean, those of us that work in a clinical lab, we definitely see, a high level of resistance. And as we have studied, if you kill the other bacteria, especially if you start killing your normal flora. So as I was reading, the article, it says that there has not been a novel class of antibiotics for gram-negative pathogens approved in over 50 years. So why is this so?
Well, it's a really hard scientific problem, is really the main answer. And 50 years ago, when the quinolones or fluoroquinolones were approved, there's actually quite a number of options between beta-lactams and aminoglycosides, tetracyclines, and then the fluoroquinolones, options that are broad spectrum that would kill both gram-positive and gram-negative bacteria. As resistance has evolved, those options have eroded steadily, and there have been next generation fluoroquinolones and beta-lactams, but there hasn't been a new class in that time, really because these gram-negative bacteria, they restrict severely the types of drugs or types of compounds that can get into the bacteria. They have gram-negative bacteria have an additional cell wall, outer membrane, and this prevents the passage of most all drugs to be drug candidates. And the ones that do get in are also subject to being pumped out. So they have very promiscuous efflux pumps. So those two things really conspire against drug discoverers, and that's the reason for the lack of success.
Okay. And so let's talk about what is Fabi? What is this mechanism of action against bacteria?
Yeah, so FABI or FABI is a key enzyme that's involved in fatty acid biosynthesis. So the bacteria use fatty acids for a whole host of operations within the cell membrane, other aspects. And so FABI is the enzyme that catalyzes the key rate-limiting transformation in that whole fatty acid biosynthesis process. And so inhibiting FABI will kill bacteria. So the compound that we found that inhibits FABI, we call Fabimycin. And so we can see that it inhibits FABI to kill the bacteria in different ways. The most telling being that when in the lab we artificially generate resistant mutants to Fabimycin, they have mutations in the gene that encodes FABI.
Okay, and then as I was reading, I saw that so there was Fabimycin, and there was also Debio 1452, and there was also Debio 1452 NH3. So can you talk about those three?
Sure. Yeah, and it's a really interesting story. The compound Debio 1452 that you mentioned is a progenitor compound. That compound is really, really potent against MRSA or Staph aureus. And in fact, a version of it is in phase 2 clinical trials against for staph infection. Staph are gram-positive bacteria. And this compound it's a really interesting antibiotic approach in that it's narrow spectrum to basically only be effective against staph. The compound does not have activity against gram-negative bacteria, but we know that the reason it doesn't have activity is not because it wouldn't inhibit the enzyme, but because it can't get into the cell. So we have been working for a number of years now on developing a general method, a general strategy or guidelines to enable drugs to get into gram-negative bacteria. And so we call these the entry rules. They're kind of 3 chemical traits that if a compound possesses, it has a high likelihood of being able to penetrate and accumulate into gram-negative bacteria. So what we did is we applied those entry rules to W1452, and we created initially the compound you mentioned, W1452 NH3, or we call it W1452 amine. And then in this paper, we kind of then use that same logic and strategy to develop Fabimycin, which is a compound that's even better in terms of its tolerability in vivo and its efficacy against the bacteria.
Okay, and you talked about activity with staff, and just for the audience, so Fabimycin. So, is it intended for gram-negative bacteria, positive, or both?
Yeah, the great thing about Fabimycin is that it also has a restricted spectrum of action. So and we show that in the manuscript, where it's really good against E. coli, which are pathogenic bacteria, Acinetobacter baumannii, also pathogenic bacteria, Klebsiella pneumoniae. And then also, and so those three that I mentioned are gram-negative infections and cause urinary tract infections and other kind of really difficult to treat gram-negative infections. The compound also remains, like its progenitor molecule, it remains quite potent against staph, very, very potent. So really, it's those types of bacteria that'll be good against. I think a virtue of it is that it doesn't kill other types of bacteria. So it won't kill, in our studies at least that we looked at, the gut commensal bacteria. And so we view this as a tremendous positive, and hopefully will be easier on patients and won't cause secondary Clostridium difficile infections, for example.
Yes, definitely. And for the audience out there, like I mentioned earlier, definitely having that normal flora, that commensal bacteria, it's beneficial for the body. And sometimes with prolonged antibiotic use, you can start wiping it, and it can cause other things. So we don't want that. So with Fabimycin, like you said, it doesn't kill other bacteria, it just kills the intended target.
Yep. Pathogenic bacteria.
Yep. Okay. And then like you said, definitely in hearing about Acinetobacter, which it tends to, it definitely tends to have more intrinsic resistance than other organisms. So you can, those can get very multi-drug resistant. So it is good to hear that this, has activity against it. So with your studies and everything, so how did Fabimycin perform, comparing it to other gram-negative, antibiotics that are intended for gram-negative bacteria? How does it compare?
Yeah. There's different ways to kind of look at that. The one of the standard ways, and this story was a great collaboration between us and here at the University of Illinois, and Walter Reed and the Broad Institute and the NIAID. And what Walter Reed did for us was they assessed Fabimycin against 100 clinical isolates, recent clinical isolates of Klebsiella pneumoniae and Acinetobacter. So they assessed Fabimycin against all those 100, and then they also assessed the fluoroquinolone levofloxacin. And so this kind of gets at your question. And it's really interesting what we show in the paper. And what you see for levofloxacin is there's a certain percentage that are sensitive, maybe 40% of the isolates, and the others are quite resistant, right? And that reflects the fluoroquinolone resistance that's in the community. Whereas Fabimycin, the range of sensitivity is quite tight, right? Quite narrow, meaning that basically all the bacteria have a very similar sensitivity to the compound. Again, suggesting that there's not intrinsic resistance to this compound out there.
Okay, yeah. So the levels of like the MIC levels they tend to be low.
Yeah.
Which for the audience, and if you're just a microbiology student or starting to learn, it stands for minimal inhibitory concentration. So can you talk a little bit about the in vivo experiments?
Yeah, sure. So the way that one advances these things is, first you look at kind of some basic tolerability So mice are the workhorse for this, and tolerability in these animals. And then look at something called pharmacokinetics, which is when you give the compound to an animal, how much of it is actually in the bloodstream and makes it to the target site. And once you have a compound that's well tolerated and seems to be distributed properly, then there's different infection models that one can look at. And there's a whole array of these. The idea is to try to mimic what the infection might be in a human host, as really trying to put the compound to the test to see if it's worthy for advancement to people. And so we looked at dye infection models, we looked at pneumonia models, we looked at sepsis models, and then urinary tract infection was a key model that we looked at.
Okay, and then so now we have mentioned the activity that it has, how it works. So what are the next steps for Fabimycin?
Well, we're hoping to push it forward towards the clinic. So we're kind of making all of the appropriate connections needed to get kind of the marshaled resources that are needed to get the data that would be presented to the FDA for what's called an IND, investigational new drug application. And so that's usually kind of a public-private partnership in some way. And then there's also, next generation versions. The nice thing about chemistry, about organic chemistry, there's always another compound to make, right? And there's always the potential that it could be even better. And so we have kind of visions about how to make the compound better, meaning more potent and better tolerated, kind of where you have a therapeutic window where the toxic dose is way different than the efficacious dose. That's the idea. And so we're actively working on that.
Okay, and anything else that you want to add about this article or any updates about this?
Yeah, I mean, I think the key thing is that I do think there'll be a new generation of antibiotics that are being developed and hopefully will come on board in the next 5 to 10 years that are microbiome sparing, gut microbiome sparing. I think antibiotics are amazing drugs, life-saving drugs. But one of the challenges with all antibiotics, really, especially with broad spectrum antibiotics, is that they do wipe out the gut microbiome, and the deleterious effects of that are really being enumerated. I mean, the obvious ones and secondary infections where you get now fungal infections or cluster C. diff infections, but also there's connections with colorectal cancer, liver cancer. There's connections with lots of other things where you get that perturbation or that gut dysbiosis, as they call it. So I think a real surgical strike on the pathogenic bacteria would be the ideal situation, right? And where one is not disrupting the gut microbiome. And it's an exciting time where we can look at these targets, and with the genomes being sequenced and all the information that's out there, we can see how the targets in, say, gram-negative pathogenic bacteria are very different than gram-negative commensal bacteria, for example. And then we can find compounds that only hit the former and not the latter.
Yes, definitely. Okay, so where can we find any updates? So you typically publish in the American Chemical Society, or do they have like an on social media feed, or that we can find more updates about this type of information, which I think is very beneficial for microbiologists to learn about?
Yeah, I think this type of work is by my lab and then labs of my colleagues around the world. There's a whole host of journals that it goes into, and I think, but ultimately the people like you and others that are close followers of this field will put it on social media. So I don't think it's really a problem anymore about people not finding these, which I think is great. I think the challenge, and a major challenge in drug discovery, is just the amount of money and resources it costs then to go to the next step, which is kind of manufacturing on a large scale and then clinical trials in humans. And so that's something that I think there's been a lot of creativity around recently, and hopefully that'll get easier and easier.
Okay, definitely. Well, Dr. Hergenrother, thank you so much for taking the time to come into Let's Talk Micro. I know that as I was reading this article, like I mentioned, was definitely, this is something that people should know about. So I accept, I definitely appreciate you taking the time to come here and talk about it. Because sometimes, like what I love about podcasts, and I tell people is that, yeah, most people, some microbiologists and people, they sit down and read an article, but then an episode, you can basically doing anything, you can listen to it and you get that information in a quick, sometimes and convenient way. So thank you so much, Doctor.
Yeah, I appreciate the invitation and also your listeners for tuning in.
Definitely. Thank you so much. Okay.
Thank you.
And that, my dear audience, is the end of this episode. I hope you enjoy learning about Fabimycin, how it looks like a promising drug for gram-negative bacteria, about how it works. As always, I enjoy sharing this information with you. Thank you so much to Dr. Paul Hergenrother for taking the time to come into Let's Talk Micro and talk about it. As always, continue bringing that passion to what you do. It's so important, remember? We do such great work. So, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.